get things going right away. So this is Dr. Bo Cleveland. Cleveland earned a JD from Boston College Law School in 1991 and a PhD from the University of Arizona in 1998. He's currently an associate professor of human development, family studies, and let's go.
Well, thank you, Danny, for inviting me. Thank you, Emma, for all your accommodations. Specifically, my son's track meet, which was scheduled about a week ago, for about an hour from now. So, thank you. And thank you, Tom, for letting me go first. Thank you, Scott. This is the first time I've been able to present in front of Scott before you because I'm always after you. So, I may go slowly. Just so you know. It's my turn. So, let's see, where's this thing? So, my talk is about using preventative intervention data sets to address the environment interactions. And it's about how to understand how to use these data sets to do this type of work. And then at the end, whether there's any sort of translational information to be gained from them. If you asked me a few years ago, I would have said no. But some of the work that we've been doing suggests that there may be some ways to apply this sort of research to translational real-world applications. I want to thank my collaborators, David Vandenberg, Mark Feinberg. They work with me all the time on this data set. They've been phenomenal collaborators. This is based upon several Prosper grants, our grants, 2 other Prosper grants, which were created by Mark Greenberg and Richard Spoth. The phenotyping was done by both Iowa State and Penn State survey research groups, and the genotyping was done by the Penn State Genomics Core. This is the overview. I want to talk about how to move candidate gene by environment research forward, how the Genetics of Prosper project does that specifically, and why it's really, really well matched for this task. I want to share results from 2 analyses related to alcohol use and smoking, and then talk about future directions and talk a little bit about our publications that we've been able to get accepted. So we've been struggling in this area. This area struggles quite a bit with how to do good candidate gene research. It's a controversial area. There's a lot of people who say this, that the work is not very strong, that it's consistent with chance. We've had quite a bit of good luck with it. To the point that I don't really think our findings are consistent with chance. But it's very important to think about the way this work can be strengthened, just like any other type of research. I want to talk about the importance of design, the importance of theory, measurement, thinking about the biological role of the gene and how that's connected to the phenotype and to the environment, and also talk about population structure. So in terms of design, in most of our data sets, these epidemiological data sets, whether they're passive correlational one-time data sets or longitudinal data sets, you really don't know if something is causing something else. Because something precedes the outcome doesn't really mean that it's causal at all. It's better than being cross-sectional, but not tremendously better. So it's really important to use some sort of design, such as a experimental design or quasi-experimental design whenever you can. This is very true for candidate gene research as well. These prevention intervention trials are fantastic for this type of research because they use randomization, which provides a lot of advantages. It helps control for selection into the environments, and we're talking about the environments here related to the intervention versus the control conditions. It also creates a great opportunity to examine these sorts of associations without regard to the type of self-selection related to genetics, such as G by E correlations. And also, they provide a lot more power than equivalently sized epidemiological designs. Some people say up to 13 times more power. That would be wonderful. But I think if it's 5 times, and I've seen that estimate more, I think that that would just be fantastic as well. We're certainly getting findings with our data set that are equivalent to much larger data sets. In terms of theory, I think these behavioral science theories and these developmental theories can be really important for directing candidate gene researchers towards relevant aspects of the environment, rather than looking at everything in your data set. So, I mean, we have a big problem in this area. There's a potential problem of these candidate genes being available in large data sets, and there's really nothing to stop people from basically just running everything. And so one of the good checks for this is basically having a strong theory to guide people towards aspects of the environment and why they should be associated with different outcomes. One of the cautions of this, however, is that although behavioral science theory can be great for directing us towards environments and outcomes, it always isn't, it's fairly vague in terms of the biological process. And so it doesn't always guide us to which genes to look at. And so it's not a total solution to the problem of capitalizing on chance. Measurement is a substantial problem in this area because after the sort of seminal findings from Caspy and Moffitt, which were about a decade ago or a little bit more, there was a rush to replicate this type of G by E in a lot of large data sets. And the larger you make a data set, the more likely you are to have poor measurement, just in general. And so a lot of these replication attempts happened in data sets that had really, really poor phenotyping. You know, asking people about child abuse when they're 40 years old, and retrospective bias, you know, all sorts of confirmatory bias. And so a lot of the strongest critiques of this area, meta-analyses, for example, were based upon, in part, some of the worst studies in terms of measurement. And family and developmental researchers are in a great position to capitalize on high-quality measurement, and this can increase statistical power to a greater extent than increasing sample size, especially when we start talking about phenotypic specificity rather than just raw power. In terms of biological role, we have genotyped probably 50 different genes, and a lot of our genes are chosen because they're the usual candidates in the area. Dopamine genes, serotonin genes. It doesn't make sense to do this and not get those genes. But at the same time, we have selected genes for their biological role and related to metabolism for specific outcomes. And so we try to do our gene selection based upon more of a biologically informed approach. So we have this sort of spectrum of considerations that should be taken into account when choosing genes. Ideally, they should have associations with behavioral outcomes. They should have associations with biochemical actions related to drugs involved, and associations with neurocognitive functioning, and associations with molecular function. So rather than just choosing a gene because it's related to something or it's one of the usual suspects, you should be able to discuss why this gene is relevant all up and down this spectrum. So we were able to get funding to add genes to the PROSPER study. It has several advantages for this type of work. First of all, it has a random prevention design. It's 14 intervention communities and 14 control communities spread out across Iowa and Pennsylvania, focusing on rural areas. Secondly, we have fairly detailed measurement of both environment and outcomes that are relevant to the process of leading towards substance use in terms of family process, peer process, and also the substance use behaviors themselves across 8 different waves. The design is a prospective cohort design. This is much stronger in terms of measurement than retrospective designs. And lastly, we are fairly well powered. PROSPER itself has more than 8,000. We have 1,000 individuals who are genotyped for adolescent and young adult analyses, 2,000 genotyped for adolescent-only analyses. We have 550 that are in the in-home sample that is incredibly well phenotyped. This is the idea that we have about how this is working. So first of all, we have the intervention influencing substance use. It's supposed to do so through influencing family context and peer context, but also have a direct effect. And we have genes, or genes, if you were, and it's supposed to have a direct effect on substance misuse. In addition, the genes can moderate, can actually have effects on family context and peer context through RGE. And then the genes should also influence the effect of the intervention, the effect of family context, and the effect of peer context. So we have a lot of different ways that we think genes can play a role. We also need to deal with population structure. When allele frequencies differ across different groups of individuals who are drawn from different ancestral environments, you can get a lot of false positives in terms of, so for example, among African Americans versus European Americans, the distribution of 5-HTP-LPR is essentially switched. So if you do an analysis looking at 5-HTP-LPR and predicting something, anything related to that ancestral group, including anything social in our country, which is a lot, you can get a false positive. So you need to control this. You need to control for this population structure. It's a classical third variable, but it's very difficult to control for if you don't have the right data. The best strategies are basically going to vary according to the type of sample you have, the sample size, the diversity, and the link between the outcome and the ancestral diversity in the population. If I have an hour-long version of this talk, and I have about 5 slides for population structure. It's sort of interesting to talk about. And we use a principal coordinate method and subsample confirmation to do this. So we use a statistical control for ancestry, and then we also rerun it in the subgroups. Although we really don't have subgroups because this is Prosper. And these, well, that doesn't work. The pink blob, those are the Northern Europeans. Prosper is from Iowa and rural Pennsylvania. So this is the distribution of ancestry in Prosper. And so this does not provide us the opportunity to actually examine different subgroups because there's really one large group and then a distribution, and then basically these different distributional sort of peninsula. And so we have some individuals who are more recently directly from Africa and more from Asia. And these are the Hispanics, which are spread out in this direction. Hispanics are very difficult to study because of the recent admixture. So when we run this analysis, we basically control for these as continuous variables, but we also rerun the analysis by cutting off the individuals who have significant non-European ancestry. This is not something that makes sense for all samples, but for our sample, when we have a distribution like that, it makes sense. So for all of our analyses that I'll show you and all analyses we do, we run all the models for everyone, and then we repeat controlling for population stratification using these controls, and then we repeat dropping for all non-Europeans to make sure that we get the same results. So I want to talk about 2 studies. The results from 2 studies. One is the genetic moderation of adolescent alcohol use trajectories and the intervention effects on the same. And the second is the impact of a nicotinic receptor subunit alpha 5 gene on smoking and intervention status. The first study looks at adolescent alcohol use trajectories And you know, as everyone here in this room knows, adolescent drinking, bad. Early adolescent drinking, worse. 5 minutes? Okay. Glad I went quickly through that. Okay. So, in terms of the alcohol study, we asked people if they drank alcohol. Because I have 5 minutes, I'll go through this quickly. We have 3 different genes that we assess with 2 SNPs each. And these are both, all these are alcohol dehydrogenase genes, so they're directly related to alcohol metabolism. They're not general actor genes actually involved with the metabolism of alcohol. And we estimate the trajectories. We look at the association between the genes and the trajectories. And then we see if the intervention changes this. So these are the findings, but this is the visual actually helps. This is the control group. This is the trajectory. So basically, alcohol use increases from 6th grade until 9th grade, and then at the midpoint, it sort of levels off, and then the increase, it increases from there, but less steeply. These slopes, from the first slope is different from the second slope, and all these are significant. So this is basically what the increase in alcohol use looks like. And then, and the ADH1C gene is related to all of these, actually just the first 2, the slope and the midpoint. And you run this in the intervention group, and what you find is that none of these things are significant. So the intervention blocks the effect of those specific genes. And we have another study looking at a nicotinic receptor. The one we use is the SNP, is known as Mr. Big because it's really, really well replicated. And we looked at high school smoking, and then the intervention versus control. This is the impact of this gene when you go from GG, GA to AA on high school smoking. This is the impact in the intervention group. So it's a nice, clean effect. And these studies basically show, one, intervention designs provide a great opportunity to examine these things. The results confirm that the interventions can reduce the impact of the specific genetic risk. What we believe is that if you really use well-characterized candidate genes that are involved in specific biological processes, you can actually have a better idea of why the interventions work and for whom. And this is, I don't have a translational slide. This is my translational point. It should be in red, I know. But years ago, I didn't think there was a real translational application for this stuff. But if we are able to find larger effects for groups of genes that are specifically involved with these biological processes and show that interventions can actually block them, I think that there are opportunities for targeting these interventions towards individuals. These are the genes we have. This is actually a call for collaborators. The ones in red are the ones that we've looked at. And we haven't looked at the other ones, so we would love to have more collaborators on this project. And we've been fairly successful with getting these publications. These are just the ones in the last few years. We've been getting them into some nice places, and so we're very happy with the results for this study. I think that's it. No, next steps. It keeps going. Oh my God. So what we want to do is we want to start to use more complex ways to characterize genetic variants. Rather than looking at single SNPs, single genes, or even gene scores, we want to start to look at genetic variants as linked to higher expression in specific neural circuits. In that way, we can use these genes to actually look at place. And that would be that's something that we want to do. And we want to start to actually do this in more sophisticated ways, just rather than adding them all up, because we really don't think adding genes up really makes sense, because we don't, as far as we know, they don't really work that way. And we want to look at how these genes interact with social networks and peers across adolescence. This study has saturated peer networks across all of adolescence. So our next grant going in is to combine the genes with the peers and look at sort of actor-partner models and genetic moderation of those processes. I think that's it. Oh, no. And we also want to analyze G by E in the context of EMA studies. Scott Bunce and I have a project, it's really his project, but he let me do the EMA on it, where we have 12 days of 4 times a day assessment of cravings and mood, and we have DNA in all these individuals that has been genotyped genome-wide. So we're going to be able to do a lot of this work more on a within-person basis. Okay, done. Thank you.
Okay, so our next speaker is Dr. Thomas Gould. Dr. Gould is the Jean Phillips Shibley Professor of Biobehavioral Health and also the head of the Department of BBH in the College of Health and Human Development.
All right. Well, thank you very much. I'm going to have to move outside of the podium because there's a few things I need to read on slides. My eyes are not that good to read the small screen there. So if I favor one side of the room, please forgive me. So my work really I'm going to talk about is a lot of things we talked about today. We're going to talk about developmental effects. We're going to talk about epigenetic changes and long-term changes due to exposure during development and mental health. But we're going to talk about this in a mouse model. I want to start off with a quote, and the quote says, "The fragile developing self-image of the young person needs all the support and enhancement it can get." So this seems like a positive statement, correct? If we look at the rest of the statement, smoking may appear to enhance that self-image in a variety of ways. If one values, for example, the adventurous, sophisticated adult image, smoking may enhance one's self-image. This self-image enhancement has traditionally been a strong promotional theme for cigarette brands and should continue to be emphasized. That's 1973 RJR. The next quote, "Our attached recommendation to expand nationally the successfully tested Meet the Turk ad campaign and the new Marlboro type blend is another step to meet our marketing objective to increase our young adult," so let's see how they define young adult franchise, "to ensure increased and longer-term growth for Camel Filter, the brand must increase its share penetration among 14 to 24 age group, which has a new set of more liberal values and which represents tomorrow's cigarette business." This is 1975 RJR. So when I first started doing nicotine research, I was in an elevator with 2 of the bigwigs. I was like wallpaper in the elevator. They didn't even know I was there. And one of them turns to the other one and says, "You know what's a real shame? Everything that we're presenting here, the cigarette companies and tobacco companies did years and decades ago." And it really is true. Why were they marketing towards adolescents? Everything we're talking about now, as if they're new discoveries, they already knew this. This is the Meet the Turk campaign, by the way, in case you are interested in what that is. Adolescent nicotine use is a major health problem. In the U.S., there's every day, more than 3,000 people smoke their first cigarette. Adolescents, more than 3,000 adolescents under the age of 18 smoke their first cigarette. Think about that as if you're a business. 3,000 new customers every single day. It's phenomenally disturbing. 9 out of 10 smokers start before the age of 18, which suggests there's something special about starting early. E-cigarettes now are replacing tobacco products, and they're being marketed as safe. But are they really safe? And you see that right now, actually, tobacco use is going down in middle school and high school students, but e-cigarette use is going way up. And based on current rates, 5.6 million Americans under the age of 18 will eventually die from a cigarette-related illness. The earlier the onset of smoking predicts more severe addiction. Adolescents are differentially sensitive to the effects of nicotine. That's been shown in both mouse, rat, and human studies. Smoking during early but not late adolescence is associated with higher rates of drug use later on in life. So we're talking other drugs besides nicotine. Smokers had impaired cognition that emerged after the initiation of smoking. So there's adolescent smokers, and they had disruptions in cognition that emerged after they began smoking. And then in children, 8 to 15, who are exposed to secondhand smoke, there's an association with depression and cognitive deficits associated with ADHD. In, 2008, DeBry and Tiffany proposed that executive function deficits due to adolescent nicotine exposure contributed to the development of nicotine addiction. So they hypothesized this, but they really didn't test this. Some of the work I'm going to talk about from my lab in the mouse supports this model. Nicotine, here's what becomes interesting, what we talked about earlier on. Nicotine produces a significant upregulation of hippocampal genes. So it upregulates genes in the hippocampus. We know that area is involved in many things, cognition, but also affect. And this effect is greatest during adolescence. Now what's important is the adolescent brain, as we talked about, continues to develop late into the 20s, all right, late adolescence into 20s. What is brain development but turning on and turning off of genes at specific times? If nicotine and other substances can turn on and off genes, perhaps it's going to change the developmental trajectory of brains that will have long-term impact. As I said, I'm talking about mice. I'm going to show one behavioral set. It's fear conditioning. We use fear conditioning because it's easy to train the animals. You put a mouse in a chamber. You give them 2 presentations of an auditory CS, so a tone, and a mild foot shock. With 2 trials, they form an association, a pretty strong association. Then the next day, you can put them in the training context. If they learned that that area, that context, was associated with the shock, they'll freeze to that context. All right, that's contextual learning. That involves the dorsal hippocampus. Then you put them in another environment they never were in, and you sound the auditory stimulus. If they freeze to that, they're freezing to the cue, and that's cued fear conditioning. This type of learning is pretty simple. You don't need the dorsal hippocampus. So if you see changes just in contextual, you know that the dorsal hippocampus has changed. If you see changes in both, you know that the amygdala or maybe the ventral hippocampus has changed. So you get brain differences in one task in the animals. So we've done a lot of work looking at the effects of nicotine in adult mice. I'm not going to talk about that. Well, I'm going to talk about it as controls. But what we're interested in is effects vary. Effects vary by the age at which someone's exposed to something, also based on their genotype. So what we looked at was 3 groups. We looked at, we looked at early adolescence, PND 23 for the mice. We looked at late adolescence, the PND 38, and we looked at adult mice, PND 53. We gave each of those groups nicotine at a dose that produced plasma nicotine levels seen in humans. And actually, it's a low to moderate dose. We gave it for 12 days, took the nicotine away, then waited 30 days. So all the mice now are adults. The adults are a little bit older, but they're not aged adults. And the early and late adolescents are now adults too. And in adulthood, we trained them up on our fear conditioning. So what we're showing over here is, on your left, the cued fear or contextual fear conditioning, and on your right, the cued fear conditioning. And what we see is that in adult mice that were exposed to nicotine in early adolescence or late adolescence, they have a deficit in contextual fear conditioning as adults. So adolescent exposure produced a deficit in adult learning. Adults that were exposed to nicotine and then trained later don't show the deficit, and it's specific for the dorsal hippocampus, suggesting that the dorsal hippocampus has been developmentally changed because of adolescent nicotine exposure. We then looked at CREB, which is a gene transcription factor. We looked at the hippocampus, we looked at the cerebellum, and we looked at the prefrontal cortex. And what we found was that in mice that were exposed to nicotine as adolescents, in the prefrontal cortex, they had increased levels of CREB, and in the hippocampus, they had decreased levels of CREB, but you didn't see the same change in adult. So you had a change in genetic factor, the gene transcription factor CREB, based on adolescent exposure, and you don't see the same pattern in adults, saying that there's a change in genetic processes. We did another experiment. We looked at mid-adolescence, but the real difference in this experiment is we looked at other behaviors too. We replicated the finding. Adolescent nicotine exposure during mid-adolescence produces adult deficits in the dorsal hippocampus-dependent contextual learning. But then what we looked at was a phenotype of depression, the forced swim test. Every SSRI drug was developed using this test. So your Prozac and all those drugs were used developing this test. And what it is you put an animal in a beaker of water, and you look at how long it takes for them to stop swimming and how long they're immobile. Basically, how soon do they give up? What we find is that for both adolescents and adults, they show a depression phenotype immediately after cessation of nicotine treatment. But the adolescents continue to show the depression phenotype even into adulthood, where the adults don't. So initially there's a withdrawal phenotype of depression. That goes away for the adults, but for the adolescents who are exposed to nicotine, that depression phenotype lasts into adulthood. So it's long-lasting depression and cognitive deficits, and the hippocampus is involved in depression. We also looked at hippocampal morphology, and what we looked at was the branching of pyramidal cells. And what we found was that in the CA1 region, adolescent nicotine exposure decreased apical dendrite branching of pyramidal cells in adulthood. So there's a structural difference, there's a behavioral difference. In addition, there's differences also in gene expression. This study, actually, you can consider this an intervention study. What we did was we had the same paradigm. We took adolescent mice and adult mice, gave them nicotine or saline for 12 days, removed it, and then put half of them on a diet that's enriched in choline. Choline is what is in spinach and other leafy green vegetables. It's very good for you. It's a precursor for acetylcholine. It's also a methyl donor. Methyl donors produce epigenetic type changes. So we gave them choline, and then we did our fear conditioning in adulthood. What we find is that choline had no effect in itself. The mice that just had nicotine as adolescents and then had the standard diet had the deficit, but choline, if you gave choline to mice that had adolescent nicotine, it prevented the adult deficit. That is interesting. But then what we did was we looked at the adult brains. We looked at the dorsal hippocampus, and we found that there's an increase in methylation in different gene groups. So if genes are methylated, they're turned off. So in the adult mice who were exposed to nicotine as adolescents, there was increased methylation. The gene group that had the largest change was chromatin remodeling genes. Chromatin is a structure in which DNA wraps around. It gives you the standard image of DNA that you see where it's tightly bound. If you change chromatin, you change how easy it is for genes to transcribe. So we see a change in chromatin-related genes due to adolescent nicotine exposure. It's in adulthood. And the animals who were treated with choline do not show this change. We examined some of the genes. We looked at two of the genes. Indeed, they showed increased methylation, and then we used qPCR to show that actually the expression of those genes were decreased. So we verified that there were changes in the expression of chromatin-related genes that we think is contributing to the long-lasting behavioral change. And these are two of the genes we looked at, and I can talk to you later about what those genes do, but they do control chromatin structure. So we think that there's long-term changes. We need to understand which genes are impacted by these chromatin remodeling. So this is the effects of adolescent exposure on adult behavior. I put this up here. Carrie Nation was involved in the temperance movement, but she did not like tobacco either, and she said that the tobacco user can never father a healthy child. So I'm going to talk about work that actually people have talked about, transgenerational work, and that really is actually a new phenomenon. We've talked about it today mostly in terms of maternal exposure. I'm going to talk about it in terms of paternal exposure, which really challenges how we think about inheritance. So what we did in mice is we took male mice, gave them either nicotine or saline, again in a dose that is a dose that smokers, moderate smokers would have as far as plasma nicotine levels, gave them nicotine for 28 days, took the nicotine away for 4 days, and then introduced them to a female. We let them mate. When the female mouse was pregnant, we took the male away, and then we took and looked at the offspring, the first generation F1 and the second generation F2. We looked at fear conditioning in these animals. we looked at the effects of nicotine on fear conditioning in these animals. So these mice never had the drug before. It's their fathers or grandfathers had the drug. What we see here is that mice whose fathers had saline, they show normal contextual fear conditioning. Mice whose fathers had nicotine, they show enhanced contextual fear conditioning. They are showing more baseline fear conditioning, which is a symptom of PTSD. When we give nicotine to the mice whose fathers had saline, nicotine enhances, which is what we normally see. Acute nicotine enhances learning. When we give the same dose, it actually decreases learning, and we give a higher dose, it still does, suggesting that the cholinergic system is altered. This is the F2 generation. We see the exact same pattern. Their grandfathers had either saline or nicotine. If grandfathers had nicotine, they have enhanced fear conditioning. Acute nicotine enhances. Acute nicotine has no effect or disrupts. The cholinergic system remains at least 2 generations. We then looked at cholinergic function. We looked in the hippocampus, dorsal and ventral hippocampus. This is basically using either nicotine or potassium to stimulate acetylcholine release in the hippocampus. And what we see is that in both the dorsal, but more so in the ventral hippocampus, that cholinergic function is disrupted if mice, or if the fathers of mice had nicotine. So hippocampal cholinergic function is disrupted. In addition, we looked at methylation. We see an increase in methylation in the ventral hippocampus. We then looked at gene expression using RNA-seq. In the ventral hippocampus, we found 952 genes that showed altered patterns of expression because the father had nicotine. In the ventral, that was ventral. In the dorsal hippocampus, we see 162. So it's actually more in the ventral hippocampus, which is an area that's more involved in fear and emotional response. And one of the pathways in the ventral hippocampus that showed the most change by paternal nicotine exposure was the cholinergic synapse signaling pathway, suggesting, again, we show functionally that the cholinergic system is altered. Here we're showing that gene expression is. And one of the genes that is changed is ChRNA3. Why do I point out ChRNA3? Well, Bo just set this up. ChRNA3 is one of a triad of genes that cluster together. ChRNA3, beta-4, and alpha-5. He talked about alpha-5. These 3 genes cluster together. Human GWAS studies have shown that that gene cluster is associated with nicotine addiction, it's associated with lung cancer, and it's also associated with other mental health processes too. And we've replicated that that gene is indeed, its expression is altered by paternal nicotine exposure. So to summarize, we see changes in fear learning. Fear learning is healthy, but when you have changes, it can be unhealthy. Too little fear learning produces risk-taking behavior. Too much fear learning produces anxiety disorders. Adolescent nicotine exposure enhanced fear learning in adults, or actually decreased fear learning in adults, but paternal nicotine exposure increased fear conditioning in subsequent generations. And so basically, not to read all these, but these effects seem to be mediated by epigenetic processes. And the paternal nicotine suggests that inheritance actually can be transmitted to subsequent generations in a non-genetic manner. And what that mechanism is something we need to look at. That is the end, but I really need to acknowledge everyone in my lab, especially Ganush Kutlo, who is the person that pretty much does everything in the lab. So thank you very much.
Thank you, Dr. Gould. So our final speaker of the session and of the day is Dr. Scott Bunce. Dr. Bunce is an associate professor of psychiatry and also serves in clinical practice at Penn State Hershey Medical Center. He holds doctorates in clinical and personality psychology from the University of Michigan.
Here we go. I work with a team. Bo wanted me to point out that he still had hair when he started working with me, so that's really important. Do have disclosure. I do have some stock in FMRI devices. I helped develop this instrument while I was at Drexel. And so I know I'm preaching to the choir for some of you, but just for the rest of you, hey, Drugs, alcohol, we have a lot of problems with that, and it's a costly problem. 185 billion per year just for alcohol, right? Big problem, probably need to address this thing. The core problem is not getting people off drugs. We've been able to get them off and separated since the 80s. That's easy. The problem is relapse. And so even though we can get them off, 50% of pretty much all drugs, up to 65, 70% will relapse within a year. Most of that is within 3 months of having stopped. And so we really need to figure out how to address relapse. So that's where we started. And what we tried to do in this project is to develop objective, brain-based measures, evidence-based measures. Now, we could say of relapse risk, but except one way you could do that is just say, okay, they're all going to relapse, because your bet would be easier. But to address, who's ready to leave treatment? Who's ready to go out and deal with this on their own with support in the community? So it's really about, can we predict abstinence? And so this becomes personalized medicine. This is how can we identify what factors are going to increase the probability that this person can go out into the community and maintain their sobriety over time. The other thing that's really important, so I'll throw out my implications for policy now, and you can judge that as we go through this, is that insurance right now pays for between about 14 and 28 days of treatment in a residential, and then they stop paying. And one reason they can do that is there's no evidence to say one way or the other whether or not that makes any difference. So they can. And if we have data that actually shows that they can do better with longer treatment, we could possibly get that policy changed. Oh, and by the way, why do we want it to be objective? Because self-report is really not very reliable in addiction medicine. Didn't surprise anybody there, right? So, what are our targets? Factors that make people vulnerable to relapse. All right, what are those? There's something known as the abstinence syndrome. And this is found in pretty much most of the drugs of addiction. Key elements, this is after we've taken them off, they're in the withdrawal stage, they're safely off of it, but they continue to have really high craving. They're really irritable folks at this point. They're not happy people to be around. There's a lot of emotional pain. There's malaise, dysphoria. They have a lot of loss of motivation for natural rewards. This has been shown in mouse models. Sue Grigson's poster talked about that. There's an increase in their reward threshold for positive stimuli. I don't know if any of you ever worked around folks that have addictions. They fill half their cup with sugar and the other half with coffee. It's like their reward thresholds are way high. And they have very dysregulated sleep. They have very low stress tolerance. And this has been found in many different drugs of abuse. So what we're going after is what we believe to be the neurobiological basis for the abstinence syndrome. Because this is, guess what? If you can't feel positive stuff, if you can't feel love from your kid, if you can't feel, like an ice cream cone's really good, what are you going to go look for? You're going back to the drug, because that's the only thing that starts to make you feel good, right? And so all of these drugs of abuse they have different mechanisms of action, but they all affect that dopamine reward system in the end. All right? And what we've learned over the last 20, 30 years, both through animal models and through some human neuroimaging models, is that as drug use goes from use to abuse to dependence, we see that downregulation of the dopaminergic system, and that affects, and there's also, it dysregulates the HPA axis, which is our stress response system, right? And it changes how the brain's prefrontal cortex functions, all right? So basically, our overall sensitivity to dopamine has decreases, and our sensitivity to stress goes up, and people are in a really rough spot. after we've just detoxed them, right? And so one of the things that Nora Volkow has argued is that the prefrontal cortex also gets dysregulated in this whole process. And that is, in her ERISA model, and I'm sure many of you are familiar with you, some of them might not be, is that we have impaired executive functioning, including our attention, decision-making, response inhibition, associative learning. Right? So we've got this combination of feeling really bad and not making very good decisions. Not great for having good decision-making once you leave treatment, right? And so we have heightened responses to drug-related cues. We know that. We have reduced response to natural rewards. We have high impulsivity and risky decision-making and dysregulation of reward and stress response system. Now, we've got a lot of really cool data from PET studies, from FMRI studies, other technologies, but how do we make that useful for our patients? Right? Well, how are we going to translate that into something that we can use to make things better? FMRI, okay, we can get some really cool data, but not everybody has a magnet sitting there, right? And if you want one, pour a bed of concrete, pay $2 million, hire a physicist, you know. Not very practical for most of our treatment clinics, right? Not next door. So, because this prefrontal cortical regulation is part of this whole addiction thing, we can use a nice little instrument called functional near-infrared spectroscopy. This was something that I helped work on while I was at Drexel before getting to the better land of Penn State. Basically, it measures the same blood oxygenation concentration like FMRI. If anybody wants a tutelage on how it works, I got lots of slides on that too if you want. The important thing is, hey, it's safe. It's 1/100th the cost of FMRI. Near zero runtime cost. Once you get a system, it costs electricity. Unlike FMRI, for instance, it's $400 or $500 an hour. Quick application time, up and running. You can pick it up, carry it in a briefcase, and that's exactly what we did. We picked it up, we carried it out to a treatment facility, and we set up shop, and we started our study after we got our NIDA grant. So I want to talk about data from two different populations. One from a group of prescription opiate-dependent patients, and the second is from a group of alcohol use disorder patients, with a focus on central nervous system measures using clinic-friendly technology, FNIR, and trying to develop objective, sensitive, and specific markers of who's going to abstain and who's going to relapse within 90 days of leaving residential treatment, right? So, both populations in residential treatment, and, well, I just got to, okay, here we are. Now, the NIDA study was a multidimensional study looking at re-regulation across 4 months. We had some people that stayed, because we work with the Cairn Foundation, I'll tell a little bit more about that in a minute. This was a self-pay facility. So some people were there for 28 days, which is standard. Some people were there for 60 days. Some people were there for 120 days. So we were able to look at some of this across time. What I'm going to primarily present to you is looking at that 28-day period that most people get. Because that's what we're going to have to work with most facilities in terms of being able to help clinicians make some decisions. And what we're measuring, so again, and this is some of the stuff that Bo was just talking about as well, is we've got not only our central nervous system measures, we also got measures of court, sleep. We did an EMA study of mood 4 times a day. So we've got a lot of measures on these folks. But today I'm going to present you these objective brain-based markers, okay? And again, we can have lots of fun talking about the other stuff at another time. So we're looking at prefrontal response to drug cue, prefrontal responses to natural reward cues, and also their response during risky decision or impulsivity tasks, okay? Again, also we had all these other measures going on at the same time. That's another talk. We worked with the Care and Treatment Center. This is a world-class drug and alcohol treatment facility. They have what they call, what we'll be talking about today, is primary care, 28 days residential. They've got extended care up to 90 or more days as well. But they have medical, they have specialty groups, they have spiritual counseling, they have family counseling. So it's a very comprehensive treatment facility with high care. And it allowed us to be able to follow these people and get some really nice data. Quickly, we started a data burst after about a week or so after they had finished their medically assisted withdrawal. We did a 12-day data burst. About the 5th day of that data burst, and this was EMA, sleep, court, these kinds of measures. About the 5th day is when we got neurophysiology in terms of our lab, all right? And so again, we've got these other things. We'll talk about those later. On that day, we looked at Q response, measured with the FMRI, in response to drug cues, in response to natural reward cues, and impulsivity decision-making tasks. Again, we did do this not only once, but we had 65 people in data burst 1. We had 21 of those go on to data burst 2. We had 10 that went out to 4 months. And then we followed them for 90 days after they left residential. Q response task is pretty standard. They had pill cues for the people that were on prescription opiates. We had alcohol for people that were on alcohol. And we also ran 3 different types of positive stimuli. They were either food, social, or positive intimate. These were all PG rated, by the way, nothing X or even R, really. So in terms of our cognitive task, we had them do the balloon analog risk task. Some of you may be familiar with that, but it's basically, it's a decision where you make a choice as to whether or not you want to give another pump to the balloon. Of course, the higher you go, the more money you make. If it pops, you lose it. So, and then we measured this with the FMRI, and we had our outcome variables were during their decision-making time and also their response to winning or losing, right? And both of those end up playing a role. So, 57 participants that finished primary care, that is 1 month of residential treatment, that we were able to get outcome data on, right? So this was done under a best estimate procedure with all the data that we could get in terms of whether or not we had good outcome. And then we had 31 age and gender match controls. Now, it gets a little bit tricky here, but I'll try and keep it simple. 26 went home after residential. So that was, and of those 26 that went home, 65% relapsed within that 90 days, all right? 31 went on to other sober living facilities. So these were step-down facilities where they had access. For instance, they would go out and work during the day and come home, or come back and stay in the facility at night. So they had access to the community, but weren't fully on their own yet. And of those, 32% relapsed within the 90-day period, okay? So we'll look at those slightly differently. Again, I'll try and keep it so it's manageable. 8 patients relapsed. So we look at primarily relapse to opiates as an endpoint, but it's also the case that many of these people, guess what? You know, some of them had some marijuana issues, some of them had some alcohol issues. So we had 8 other people who didn't relapse to an opiate, but did relapse to alcohol primarily, okay? So we didn't want to include them as not relapsed. And so we actually do we have some analyses where we've got them in as relapse, other ones where we just look at the people that have opiate relapse versus not. That makes sense? So here I'm going to show you a series of slides in terms of some of the outcome with the FMRI. The thing that I want you to be aware of is that in each of these cases, what you'll see is that the relapsers differ from the abstainers and the controls. Controls and abstainers look roughly the same. Relapsers look different. So this is the response to the pill stimuli. And this is our relapse in terms of treatment outcome, 90 days, relapse to any particular drug. And again, mostly that's opiates and alcohol. Abstainers, controls look the same, relapsers differ. Same, this is their brain response to positive social stimuli. Same pattern. Relapsers differ from abstainers and controls. In terms of the risky decision making, patients relative to controls had fewer winning trials. Not entirely surprising there, but abstainers and relapsers did not differ in terms of their actual behavioral data. But in terms of the neuroimaging data, you can see that the reaction to wins, reactions to losses, and the decisions during which they win all differed between those who abstained and those who relapsed. And you can also see that the reaction to loss and reaction to win are in opposite directions. All right? Now, what we did was combine the data from response to drug cues, response to natural reward cues, and response during the BART task. And what we were able to show, if we just look at the overall group, take everybody lumped together, we got about an 83 to 84% accurate prediction of who was going to relapse versus who was not going to relapse at 90 days. Okay? And you can see there our sensitivity is a little better when we're looking at any substance. It's a little bit different when we're looking only at opioids. Part of that is because these measures are probably not specific to opioids. The only thing that's a little bit different if you're looking specifically at pills versus not, but in terms of response to positive stimuli, response to the risky decision making, that's the same whether they're addicted to alcohol, whether they're addicted to opiates, et cetera. Okay? So these are non-specific responses. If we look at splitting it out by the sober living versus going home, again, we're right in the 83 to 84% accuracy in terms of correct classification with a binary logistic regression. And when you look only at opioid, we're even a little bit better. We get up to 100%. I don't believe that figure to some degree because we're overfitting the model a little bit probably. But still, that's a pretty darn good outcome in terms of predicting treatment outcome with prescription opioids. Now, what do we find? A combination of prefrontal cortical responses to pill cues, natural reward cues, risky decision, correctly classified 90-day relapse to any substance with about 83 to 84% accuracy. That's pretty decent. 80% is cut off for starting a clinical trial in any kind of med. All right, so we're there. We get very similar results for alcohol dependence. We looked at, and this was research done, Jonathan Harris was my graduate student. He was actually hired by the Care and Treatment Foundation, so he's working out there now. And he looked again at an alcohol population with alcohol cue response, natural reward cues, and he used the color word Stroop task rather than the BART. And I'll differentiate that. He had a sample size of 50 patients. Of those, 32% relapse, so 18 out of 50. Six of the relapsers were females, 32 were abstinent, and 7 of those were female. So the Stroop, I think most of you know this, but basically it's how fast that you can read those. You have to say the font color rather than reading the word. And when you get from going where they match to where they don't match anymore, all of a sudden you slow down a little bit because it's a little harder. You want to say blue when it's green. And so that's the Stroop, for those of you that aren't familiar with it. With the Stroop, what you can see again is the same pattern where relapsers look different from abstainers, and abstainers look like controls. So we see that same pattern. Now, the actual Stroop performance did not differentiate between abstainers and relapsers, but the neuroimaging did. And again, you're going to see the same pattern. Go dog. There we go. Okay. Same pattern in terms of response to natural rewards. Relapsers differ from abstainers and controls. And basically the same pattern in response to alcohol stimuli. Again, relapsers differ from abstainers. Abstainers look like controls. Put them together, and again, into a logistic regression, correct classification of 84.8%, and with a leave one out analysis, still at 85%, and sensitivity and specificity is right there where we want it in terms of being ready for a clinical trial. And again, for those of you that don't play with this stuff all the time, sensitivity is identifying relapsers as relapsers, and specificity is when you identify abstainers as abstainers. Okay? So, conclusions. All right. This is very promising. I'll still say that it's somewhat interim data, but it's very promising data, and the fact that we can do it in 2 different populations as well. We've got a combined PFC activity, 84 to 85% accuracy, with the combination of those 3 types of responses. We think it probably represents a general vulnerability to relapse. It probably should also then generalize to, for instance, cocaine, marijuana, some of the other drugs of abuse as well. We don't see any reason why it shouldn't. What's really important, we demonstrated clinical utility. We were right at Karen running this in an office, and we were able to do it without taking anybody out of their treatment. Right? We did it during times when we had access, they had downtime, et cetera. So everybody stayed in their treatment, stayed in the residential, and again, the overall cost was, now the study cost money, of course, but in terms of the potential to put this in place at various treatment facilities is very, very possible. But we need to replicate it to determine overall reliability. And then ultimately what we should do is develop a best fit model. How can we optimize this? How can we make this as streamlined as possible? One of the other things that I believe is that probably this is a good predictor of relapse at 90 days. Is it a best predictor for a year? Maybe not. Maybe not. There may be different factors that are playing at 1 year out or 2 years out than there are at 90 days. But if we get them past this 90 days, that's a really good thing. And it gives the clinician is making the decision at the time of saying, okay, is it time to go home from residential, or do you need to go to a higher level of care? If we had some objective measures, when you go in and say, doc, you know, I know my lipid level's really good, I've been good, you know, and he says, oh, good, that's good to hear, I'm glad you have great, no. He says, come here, take your blood, like, here's your number, right? We don't have that in psychiatry. We don't have objective data like that. And this would potentially provide an objective measure that could say, yeah, this person looks like they have a pretty reasonable possibility of going home. One last tidbit. We had a patient, EF, in our alcohol study. He's a 48-year-old male, started drinking heavily in his 20s. He opened his own architectural firm at the age of 24. He actually was making it 6 figures in his business. He was diagnosed with lung cancer at 31, which he beat. Okay, so he had been declared cancer-free. In his late 30s, he started drinking 2-day binges, primarily nice wine. Started drinking up to 3 bottles of wine per day. And so he came into treatment. And all of his clinicians thought he was doing great. He was a good person. He was very sociable. He was working his steps hard. He felt very positive. So we went home, and in 2 months, his brother called, and he had died of alcohol poisoning. And we said, you know what? Could we have known this? So we went back, and we looked at his numbers, and when you looked at his craving, all right, his self-report, how did his craving do? He said, you know, I'm seeing these alcohol cues, I'm not craving. Good, right? What happened to what about his severity of alcohol dependence? Well, he had been kind of high on that, right? He'd been a little high on this. But if we look at his neurophysiological markers and the Stroop, he was right in there at the mean with the relapsers and nowhere close to where the abstainers were. He was quite high in terms of his response to the natural work cues, so very different from the abstainers. And in terms of the alcohol cues, again, each of these physiological markers put him in a category that could have told a clinician he's not ready. He believes he is and he wants to be, but his brain tells us not. This is the potential for this kind of technology and this kind of measure. So, yeah, we have a possible measure that does need further development. We believe it's a potential biomarker of early relapse, although not necessarily long-term recovery, but we need to replicate it. We need neural response over subjective measures, which are a lot easier, a lot cheaper, believe me. And we need to develop this into a streamlined clinical application, that is an optimized, minimized protocol. Impact on policy. We want to be able to have third-party payers say, when the clinician says, this person really needs to be in treatment for another 30 days, is to be able to get that paid for. And right now we can't. Right now, 30 days, 28 days, you're done. And you don't have any, it's because we don't have the data. So that's.
So we'll open up the questions. I guess in my role of moderator, I'm going to ask the first question. This is oh, sorry. This is directed to Dr. Gould. You mentioned use of choline, right?
Yes.
As a sort of a substance that can potentially reduce the methylation.
Yes.
Right.
Yep.
Any thoughts on using sort of choline as a sort of a prophylactic kind of supplement in diets to help reduce this sort of transgenerational effect in MSC?
Yeah, so the transgenerational effect and what may prevent that is really a great question. We haven't even begun those experiments, but one thing would be, well, let's take a look at choline and see if that makes a difference. And one of the things we want to do right now is actually also try to understand what is changed that allows transmission across generations. I mean, that based on our understanding of genetics shouldn't happen. And so, so there's two things we want to do is understand what's driving that mechanism, but also look if we can reverse it. And one of the approaches I like to use is if you reverse it with something, that may give you a clue as to what to look at. So, and there are studies that have looked at in animal models, for example, a neonatal exposure to alcohol, that choline can reverse some of those deficits. So choline may be a mechanism.
No, so tag on to Chuck's question. So do you think, so you mentioned that we get choline from leafy greens, and so you would assume that those who have a diet with better nutritional quality may have diets that contain sufficient choline. Do you think that there's a potential mechanistic explanation in dietary choline for differences that we see between low SES smokers and high SES smokers, in that, this thing that they try to quit as often, but low SES smokers are like 3 or 4 times less likely to successfully quit? And do you think that that's potentially a mechanism?
I mean, it could be, and again, it's the developmental component too, and whether choline can help with it. I know there's some studies, Sherry Leonard had been doing them, looking at actually choline supplement as far as Alzheimer's disease and seeing some effects. And so it's possible that in the diet, but what we really need to look at is what is the concentration in the diet too, and also the other components of what they're taking in that may interfere with actually the use of or the metabolism of the choline too. So in the mouse, it's really easy, unfortunately, because we supplement with choline and we control everything else. But actually, yes, if you could actually do those studies and look in humans, or actually just supplement and see if there's different outcomes too, I think that would be worthwhile.
So that's great work, first of all, and it's intriguing. I'm wondering, though, when you have a comparison between abstainers and relapsers, you have a natural confound of the recency of drug use. And so, given your message...
The recency of drug use.
Of drug use. So you have abstainer people who are not using. And so, pardon? Oh.
The abstinent relapse is out from the data position. Right?
It's 90 days, so it's 90 days out.
So we measure
those
them.
That are abstinent.
So they withdraw. We do the measurements. They finish their residential. 90 days after their residential, we're tracking them for those 90 days. So in those 90 days, we get a decision of abstinence or relapse.
During that whole 90-day period.
Yes.
And then you have the relapsers who have used during that 90-day period.
Correct.
And so isn't there a confound in the potential for that to affect some of your brain measures?
Well, the brain measures are done.
Oh, they're predictive.
Yes.
You're not looking at
Yes.
change.
Well, we did have a group that did go out further, that subgroup, but we started their clock for 90 days after they left, and then we've done other analysis where we've controlled for the amount of time they've been in. It was a subsample, and so yeah, so all of this is, all the measurements were done before them leaving treatment, and outcome is after.
Is there any indication that after the 90 days, something like that, that the same markers change or look different? Like if they're doing the bark task again, or if they're doing, or the response to cues. In other words, those who are abstaining, do they look different from relapsers later?
Well, we're still in the process, because we have 3 data sets of neuroimaging data, we're still processing some of the second and third out. Now, the thing is that the people that we have those measurements on have been in residential treatment for that full time. So to some degree, yeah, what we're looking for instance, we know in the court, which is a lot easier to process, in court, we see a change across those 3 data bursts such that by the time we get out to that 4th month, they're the same as controls. Whether heightened court at 1 month, at 2 months, and by month 4, they're about the same. We haven't fully processed the data imaging for month 4 yet. And unfortunately, it's a fairly small sample, but we don't have, after they've kind of left and gone, after they've left residential, we haven't gotten them back in to be able to answer that question.
Other questions? Okay, so join me in thanking our...
So we are reduced in numbers, and it has been a long day, so we made the executive decision to not do the breakout session. But what we really are going to do is find an effective way to follow up, because one of the things that we have achieved, certainly one of our goals, I think, today, in showcasing some of the really amazing work that people are doing. And I have to say, like, all of the speakers knocked it out of the park. I think you all did a phenomenal job, and I am so grateful not only that you came here today to speak, but for all of those of you who have already participated in the intro, and those of you who haven't. Thank you for your effort and your support. But yeah, it's been a really awesome day. But what we'll do is we'll follow, because one of the things we had wanted to do was to try and generate some novel collaborations. There's been some negotiation with SSRI about some potential funds for pilot projects that will be kind of P-TRAN. They still have to go through SSRI's normal review process, but we would really like to push that agenda, so we will reach out to everybody who came and see if we can find some way of making those collaborative relationships happen. I do just want to say some other quick thank yous, even though most people have gone. I want to thank SSRI and CTSI for giving us money to be here and for all the food. Oh, and if anybody would like to take snacks home, cookies to children, or pretend that cookies are for children, feel free. I want to thank Denny for her leadership. I was going to say for bringing me back from Europe, given the current administration, I'm not so sure, but overall I'm glad to be here.
Sorry.
They're out. Oh no, Lee's here. I want to say a huge thank you to Lee, who is ignoring me. It really wouldn't have happened without her. She's been a huge, huge help to me, and I'm very grateful. She just probably got roped in because she's in the office next door to me. To Mel, to Charles and Jess, who are also outside. Jess is on the phone. Everybody wave at Jess. Again, to the speakers and the moderators, everybody who's participated in P-TRAN, and also any poster presenters who are still here. Thank you as well for presenting your work and being part of the day. Denny, do you want to say?
I'll just say that we're looking forward to any collaborations that arise, and we'll probably be able to get some seed money for that, so that if people want to get together, I'm already wanting to collaborate with about 5 people. But hopefully that's what it will lead to. Maybe next year we can present some preliminary data from some of the pilot work that's been done. And I also want to thank you all for sticking it out. It's been a very long day, and those that didn't stay will be severely punished. So let's all go and enjoy your evening, and thanks again. It was really great.